Okay, so we're picking up where we left off yesterday, which is on slide 16, and we're going to go through slide 33. So we ended here where we talked about connective tissue wrappings yesterday. We talked about how each fiber, if you remember, that's like your spaghetti noodle, is wrapped in a casing called the endomysium. And then all those fibers are bundled together into something called a fascicle, which has its own binding called a paramysium. And then all those fascicles are put together to create a muscle, which has its own wrapping called the epimysium. Okay, so for today, remember that muscles have tendons. So those epimysia, the outer covering, blend into strong cord-like tendons that attach muscle to bone. So if you remember yesterday with the saran wrap, that tail that came off the end of that epimysium, well, that was the tendon, okay? Tendons are composed of fibrous connective tissue. Fascicle arrangements, okay, so fascicles are arranged parallel. They are circular, convergent, which means they come together. They're unipennant. They're bipennant. Well, they can be unipennant or bipennant. So that means that they can come together. As you can see here is an example of bipennant, where they there's two that come together. There's a multipennant muscle where three come together to form one muscle, and then there's a unipennant where there's one stretch of muscle that comes together. So that's what it means by unipennant, bipennant, or multipennant. Okay. Some of them are parallel. So like, for example, the muscle you made with the spaghetti noodles is parallel. All of the muscle fibers are running in the same position. And then there can be an example of parallel fusiform, which means they run parallel, but then they attach at one point or the other, like your biceps. Okay. Origin and insertion. The origin is the attachment to the immovable or less movable bone. So in this particular example, the origin is up here in that ball and socket joint. Okay, it's attached to a more immovable point. We call that the origin. The insertion is the attachment down here on your radius and your ulna. That's a more movable bone. That's the bone that contracting your biceps causes that bone to move. That's the insertion point. When a muscle contracts, the insertion moves towards the origin. Okay, so the muscle shown here is your biceps brachii. It has two origin. That's why we call it the biceps, because bi means two. One origin is at the coracoid process of the scapula back here, and the other is at the supraglenoid tubercle of the humerus right here. So you can see that there's two strands, right? One attaches on the humerus and one on the scapula. So it technically has two origins, which is why it's called the biceps, two. Okay. The insertion, there's only one insertion. You can see there's only one strand here that attaches onto the radius and pulls that radius up when you contract your biceps. Muscle names. So skeletal muscles are named based on a bunch of different factors, and that could be the direction that the muscle fibers are going. It could be the relative size of the muscle. It could be the location of the muscle or its origin, right? So biceps, because it had two origins, it was called the biceps. Okay. It could be the number of origins, like we just said. It could be the shape of the muscle or the function or the action. So the names of muscles depend on how they're oriented, how big they are, where they attach, how many times they attach, their shape, or what they do. So the muscle names vary a lot. Some examples of names. Okay, so the gluteus maximus is the largest muscle of its group. There's also a gluteus minimus and a gluteus medius. So maximus would be large. The gluteus maximus is the largest muscle of its group. The gluteus minimus, mini, would be the smallest of that group. And the gluteus medius would be in between the other two muscles in size. Another example is your biceps. The biceps brachii is named for two features. Biceps refers to the fact that it has two origins, like we talked about earlier, and brachii refers to the muscle being in the brachial area. If you remember when we were talking about brachial areas in our terminology in Unit 1, right, that refers to your arm. So the brachii refers to the fact that it's in the brachial area. Biceps refers to the fact that there's 2 points of origin. Here is a list of some of the anterior skeletal muscles. So if you want to take a look at that, you can pause the video or you can open up the slide through Canvas. You might recognize some of them, like the pecs, the pectoralis major, right? Traps up here, deltoids, biceps. These are generally really well recognized muscles. The posterior or backside, your triceps are usually pretty easily recognized. Those are on the backside of your arm. Gluteus maximus is usually remembered pretty well. So I would take a look at these because you are going to have to label these on a diagram that you'll start working on today. And for the lab practical, you'll be given a diagram that you have to label. Okay. So I want you to think of examples where two things move because of one another, because that's how your muscles move. So most body movements are the result of two or more muscles acting together or against each other, which means that whatever one muscle does, another one can reverse. So I want you to think of some examples where there's, a prime mover and an antagonistic or against muscle pair. So like if you think of your biceps and your triceps, you have a prime mover, okay, or an agonist. Those mean the same things. That's the muscle that produces a particular movement. So that would be like your bicep. If you flex your bicep, if you curl your bicep, okay, the antagonist produces the opposite effect on the same bones. So if you contract your biceps to make your radius and your ulna move up, then you'll feel the tricep on the backside of your arm does the opposite of what you're doing. If you relax your bicep, your tricep does the opposite, and it produces the opposite effect on the same bones. Because if you think about it, if your bicep and your tricep were both pulling on the bone, it wouldn't go anywhere. So one has to do the opposite movement of the other. So here is kind of a picture of your bicep and your tricep. Your bicep is on the front side of your arm. The triceps are on the back side. Your bicep is the prime mover or the agonist when you flex your arm, and the triceps is the antagonist. So you can see in this picture, when you flex your arm and you use that bicep muscle to pull your arm upwards, your tricep relaxes and it's not as flexed. And then when you relax your arm and you let it hang back down, your bicep relaxes and your tricep engages. Then you have what's called synergists. So synergists are muscles that help stabilize a movement. They either produce the same movement or reduce undesirable movements. So like if you're picking something up, your biceps will produce that movement, and then your brachioradialis, which is the muscle attached to your radius, when you flex your forearm, you can feel it, is one that helps stabilize that glass of water as you lift it up. You also have something called fixators. Fixator muscles stabilize the origin of a prime mover. So for example, when you bend over to pick something up that's heavy, fixators in the trunk of your body will help you maintain your balance so that you don't fall over. So fixators stabilize the origin of a prime mover. If we dive a little bit deeper into the microscopic anatomy of our muscles, okay, we know that each muscle fiber is a cell. If you remember from yesterday, I said muscle fibers and muscle cells, same thing. So each muscle fiber is a cell, okay? A sarcolemma is the cell membrane. Sarcolemma is the cell membrane. So that's this right here in this picture. The fibers are made of what's called myofibrils. So this is all one big fiber, okay? These inside pieces right here, like the one that's jutting out, is a myofibril. Mitochondria, remember mitochondria are the powerhouse of the cell. Mitochondria is what supplies the energy for your muscles to contract. So if we continue to look at the cells, okay, or the fibers, they're stimulated by electrochemical signals. So those are the signals sent from your brain, and it travels along the membrane or the sarcolemma right here, okay? Then you have something, if you zoom in even further, You have these T tubules, okay, these little pink things that carry the signal to the muscle fiber. And then this wrapping, looks like netting, is called the sarcoplasmic reticulum, and it stores calcium ions needed for contraction. So yesterday when you guys were asking a question about muscle cramps, we talked about how your muscles need certain ions to function. Calcium ions are one of those ions that your muscles need. And this webbing looking stuff, the sarcoplasmic reticulum, stores the calcium ions that your muscles need in order to contract. Okay, so we talked about myofibrils, right? Myofibrils are these things right here that make up a muscle fiber. Myofibrils are long, ribbon-like fibers of a muscle cell. Myofilaments are thread-like proteins in the myofibril, okay? So the myofilaments make up the myofibrils. Myofibrils make up the muscle fibers. Muscle fibers make up the fascicles. Fascicles make up the muscle. Okay, so if you've ever seen those Russian nesting dolls, that's kind of what this is like. There are two kinds of myofilaments. There's actin, which is a thin filament, and there's myosin, which is a thick filament. Okay, just a couple more slides. There's something called a sarcomere. A sarcomere is the bundle of muscle fibers made of actin and myosin that function together. So this is a picture of a sarcomere. You have a worksheet that you'll work on today with labeling the sarcomere. Okay, thin actin filaments shown in this picture in light green and thick myosin filaments shown in this picture in purple will bind together during muscle contraction. So when the muscles contract, these thin green actin filaments will bind together with these thick purple myosin filaments. Okay. Myosin molecules, shown here in purple, have a club-shaped head that will extend toward and bind to the actin, which is in gray. Okay. Actin contains myosin binding sites, so think of it like a landing strip for an airplane, and only that airplane fits in there. Actin contains these sites where myosin can bind, and they're covered by proteins shown in green until calcium ions are present. So if these calcium ions aren't there, this myosin can't bind here. Once the calcium's here, now myosin can bind. Okay. So just kind of a heads up about what's going on in here. Tropomyosin is the green ribbon in this diagram, so it's labeled here. And that's just a long protein that wraps around the actin and it covers the sites where myosin can bind. When calcium ions, shown here in yellow, are present, another protein called troponin, right here, moves the tropomyosin away. So once this is present, this starts to move away and myosin can bind. Until calcium is here, this whole stretch of green will block the site for myosin to bind, and then it can't move. Okay, so let's look at some of the structures to review. Okay, so you have your muscle fiber. This whole thing is your muscle fiber, okay? This is your muscle cell. These little things are your nucleus, okay? Then you have myofibrils. So your myofibrils are right here. That's this part, which if we zoom in, is this whole thing down here, okay? So myofibrils is the part of the cell that lets you contract. Then you have the sarcomere, okay? And the sarcomere is these things in here. And you have your actin and your myosin, okay? Your myosin is these thick red bands, and the actin is the thinner blue bands. And when your muscle contracts, these two things will hook together and pull on this myofibril, which pulls on this muscle fiber, which pulls on the fascicles, which pulls on the muscle, okay? The banding pattern here, these light and dark bands, this is caused by an overlapping of these filaments in here, okay? That's why the two types of muscles that are striated, cardiac and skeletal muscle, have these. Smooth muscle doesn't have these overlapping bands, so you won't see this striated dark pattern. Okay, so that's it for today. We're not doing any more. That's all we've got. If you have any questions, feel free to email me. or ask them when I get back on Wednesday. Or I don't have you Wednesday, so it'll be Thursday.